This video will show you how to use the NCBI database and Primer Blast to design PCR primers from sequences from the NCBI Nucleotide database. To get started, navigate to ncbi.nlm.nih.gov/nucleotide. Alternatively, you can go to any NCBI URL and select from nucleotide from the pull down at the top of the page. Once you're in the nucleotide database, you can enter then the accession for the sequence that you're interested in. This could be an accession number that you've seen in a paper from the literature. It could be something that you've identified from elsewhere. It could be something that you're copying and pasting in. There are alternative ways to access Primer Blast. You can go straight to Primer Blast from the NCBI, but I find if you go this way, then you can be sure that the nucleotide sequence that you're going to be working with is the one that you're really looking for, and you haven't entered the accession number incorrectly. So you can see by doing search that this is an acanthamoeba sequence. It's of a hypothetical protein, and when you scroll down, you can see all of the header information that has gone into the NCBI database, the authors who have contributed to the paper and the publication from which this nucleotide sequence has been entered into the database, and this predicted translation of the hypothetical protein. One of the tools that you can use when you are here on this page, you can run BLAST, which is popular tool to use here, but you can also pick primers. So this is what we're going to do today. We're going to pick primers under the Analyze This Sequence on the right-hand side of the page. From time to time, the NCBI web pages may change, so this may look slightly different from the video that I'm recording today, but that's fine. Navigate through, and all of the basic information that NCBI provides will still be there. It may just look a little different. So today we're going to pick some primers for this sequence. It can be any sequence that you're interested in. It doesn't have to be eukaryotic microbiology. It can be a human sequence, something related to plants, something related to bacteria or viruses. It will work all the same. And this takes you into the Primer Blast environment. And you can see here that rather than taking in the sequence, it's taken the accession number. So as I mentioned before, you can go straight to Primer Blast. You can just type in the accession number here, but then if you had entered this number a little incorrectly, or you'd been given the wrong number, or it was a typo in the original paper, you wouldn't necessarily know that this wasn't correct. So by going to the nucleotide page first, you could verify that you're looking at the right thing. You can also paste in the sequence here. So I could have copied the sequence from the previous page and pasted it in here. But the NCBI folks have given us a straight link so that you can just take it across. Okay, so today I'm going to set the PCR product size minimum to 900. So let's see where we're going to do that. PCR product size minimum. So it's currently set at 70. We want to set this at 900. Okay, so that will be a minimum of 900 bases. And I want to set the number of primers to return as 1. So this is here, number of primers to return. Currently it's set at 10, and I want to change this down to 1. I only want to see 1 primer pair. I only need 1 primer pair. I'm going to set it down to 1. Now, oftentimes you want to see more than one set of primers because you want to decide for yourself which primer pair you might like to use. but today we're just going to do one. The next thing you often want to check is that these primer melting temperatures are what you would like them to be. So make sure those are set where you would like. Sometimes this default is fine, and today I'm going to leave that as the default. That's fine. And we can continue scrolling down, making sure everything here is set at the default is often correct, but that may not be what you want to use for your particular project. The next thing I want to change here is the database, and I want to set this as NR. NR is the non-redundant database. So that's what I'm setting there. And everything else, scrolling on through, this is all fine. So I'm now going to push Get Primers. And this is then the primer design tool, which is based on primer 3. This is going to compute where the best primers are in that DNA sequence. Now, depending on the time of day and how much else there is that the server is needing to do, it can take some time for these primers to come back, and you can keep checking how long it's going to take for your primers to come back. But they should be back in a fairly rapid amount of time, and it will keep loading. So that's what just happened there. The primers will keep loading and keep refreshing. So you don't have to keep refreshing yourself. You might do it anxiously, but you could just leave this and start a new tab, maybe check for some other primers, do some other analyses, check some emails, and then come back to it. And rest assured, there will be some primers for you. It will tell you as it progresses the time since your submission, so you can check how long it's taken for your primers to be generated. And that will keep coming up there. And we're just going to sit and wait for our primers to be generated. It's been 74 seconds now. And what we're looking for is a forward primer sequence and a reverse primer sequence to come out of this analysis. It's going to use, as I mentioned, the primer 3 program is behind this, which will find the best primers with those annealing temperatures. It will check to make sure that those primers are not going to form hairpins that will anneal back on themselves, which will be adverse to making a PCR product. It will make sure that those two primers are not going to self-anneal with one another. So the two primers will join against one another, which would be adverse to making PCR. And with the blast involved, so it's not just primer 3, it's primer blast. It is also going to check to make sure that those primers are not going to hit elsewhere and then generate a sequence that you wouldn't expect. So when you run the primers, having generated PCR out on a gel, you wouldn't get a second band that you didn't expect to see, or maybe a third or a fourth band, because that is very frustrating. So here we have the results have come up, and it says at the output. So maybe if you went straight to primer 3 and you just put in your accession number, just check here to make sure that this is what you're expecting. The genus, the species, what gene you're looking at, just to double check. And then primer pairs specific to the input template. No other targets were found in the selected non-redundant database, so that's great. And you can actually specify this down to just be the organism that you're looking for. You can specify it to whatever you need to for a database. This gives you a graphical view of the primer pair, and you can see primer 1 is here facing in that direction. And if you hover over, you get detailed information there. And primer 2 is here. So it's in the reverse direction. It's going to anneal on the other side of the DNA strand, and that will then generate your PCR product. And there's some more information down here. There's the forward primer and the reverse primer. These sequences are shown 5' to 3'. And here's your product length, 957 bases. So that's how long the PCR product will be when you run it out on a gel. Here's their length of each primer, 20 bases, and you can see they're the same size, 20 bases, where it anneals in the sequence that was from the NCBI, and then the melting temperature. So they're bang on the same temperature. So that's great as far as generating a great PCR, that your two primers are going to be at the same temperature. Okay, so you can see how that works out. You can then copy and paste those primers out into your lab book. Make sure you've got them recorded in your lab book. Make sure you know the melting temperature, the percent GC, where they stop and start within your gene or your sequence of interest, and then you can get them ordered and you can start working with them in the lab to generate PCR products just as soon as they arrive from the manufacturer. Okay, I hope you have found this video useful and that you're able to use it in your own research. You can stop and start it as you're doing your own primer blasts anytime you'd like. Thank you very much for joining me, and keep watching more videos, and please do buy my book, Bacterial Genetics and Genomics by Lori A.S. Snyder, available at all good book retailers from CRC Press, Taylor & Francis.